Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be moving on to the third part of this module of soil bacteria as stress evidence in the course of forensic microbiology. So again, before delving into today's session, I would again like to mention the same thing again or repeat the same thing again, that as you can see, this is the third part of this particular module, soil bacteria as trace evidence. I think this will be the last lecture of this particular module. After that we will be moving on to the next module. So I think this will be lecture 39 or 40 of this course of forensic microbiology. So since before this I have already covered 2 lectures of this particular module, so if you have not yet watched those 2 lectures, or maybe if you have forgotten what I actually explained or discussed in the previous 2 lectures, then please go back, watch those lectures first, and then come back to this one, because otherwise what will happen if you do not have in mind that what I explained or discussed, particularly in the previous lecture, that is where I discussed the second part of this particular module, you will not be able to understand and keep track of what I am discussing in this session. Okay, so therefore it's my recommendation and like earnest request that please go back and watch those lectures first and then come back to this lecture. Okay, so without wasting further time, I will delve into today's topic. So the first subheading that I will be discussing in this session is feasibility of next generation sequencing, that is NGS, for forensic soil analysis. Okay, so now what do we mean by this word feasibility? Feasibility is the ability to carry out a task. That means how easily that, or how effectively, how effortlessly rather, a particular task, which in this case we are talking about NGS, that is next generation sequencing. So how easily deliverable is this part of NGS as far as forensic soil analysis is concerned. So that is what we mean by feasibility. So when a particular task or when a particular function is having very low feasibility, that means it's very difficult to carry out that task. Difficult as in with respect to various factors, like it may be very time consuming, it may be very tiring to do, it may be a very tedious job. Okay, or maybe it is very costly, it is not at all cost effective, maybe it is not at all friendly also. Okay, it is very much environmentally non friendly, that can also be a reason for low feasibility. Okay, or maybe it always needs very much of expertised people, which is again indirectly costly, isn't it? Because if we want to hire, or if we need to hire a bunch of expertised people to carry out a function or to carry out a task, then obviously we have to pay them accordingly, we have to pay them on the level of their expertise, right? That is again obviously indirectly costly. So these are the different factors on which the feasibility will be decided. So if it's low feasibility, that means it's very difficult to carry out that task successfully. And if something is having a very high feasibility, that means it's very effective or very effortless to do that function or to do that task effectively. Okay, so let's move forward and let's see what they're discussing over here. Next generation sequencing is clearly the most promising microbial profile generating method available today, producing massive data sets with higher resolution than older techniques. So what they're saying over here, that the resolution is much higher than the older techniques, means the conventional or the traditional techniques that are still now followed, but they are not that much rewarding. Why? Because they are not giving us higher resolution. Higher resolution means what? Higher resolution means higher accuracy, right? Means more accurate, more precise results they will be giving, more precise conclusions, more precise inferences can be drawn from them. Okay, so higher resolution will be a very important point over here, which I would like to highlight for you. Okay, moving forward, so there was a scientist called Sensorborg, and he did this in the year 2009. 2009 means, just imagine, it's around you can say 25 years ago, right? No, not 25 also, it's around like almost 30 years ago. 2009 means. Okay, What he did was, Sensaborg, he was a scientist or a researcher. He recommended three qualities that a new microbial methodology for forensic science should possess before it will be accepted for widespread use. So what are the three qualities? Three qualities. So let me highlight first that. So we need to discuss about three qualities which was highlighted by Sensaborg. So first was the methodology should result in reproducible data sets. Second quality was the microbial profiles generated using the technique must reliably allow for the differentiation of samples with unique origins. Means we need to classify or we need to categorize the different types of microorganisms on the basis of their origin. Okay. And the last or the third quality is the data analysis methods utilized should be objective, identifying differences or similarities between or among samples while also providing a statistical measure of association. So statistical measure of association, that is again a very important part that we need to highlight. Why? Because I think in the previous lecture also, when I was discussing part 2 of this module, again if you go back you will be able to find it out, I discussed about the statistical significance. Okay, particularly I discussed about the p-value, that the p-value needs to be less than 0.05, then only we can say that the study is statistically significant. Okay, so that thing comes here also, that no matter what studies we are doing or what kind of analysis we are doing, whether it's next generation sequencing or high throughput sequencing or any other kind of tech savvy machine learning based tests that we are doing or analysis that we are carrying nowadays, we need to provide a statistical measure of association. So this is something again that I would like to highlight for you. Statistical measure of association means we need to establish the statistical significance of our findings. Okay, moving forward, determining whether microbial profiling of soils fulfills these recommendations requires exploring several factors that might affect the profiles produced. Okay, so the next subtopic that we will be discussing in this session is differentiating diverse and similar habitats. So that is again a very important part, because ultimately we are talking about soil mycobacteria, soil microorganisms or soil bacteria, right? How they will be helping us in forensic analysis, or how they will be acting as trace evidence. That is the main topic or main motto of this module, right? So whenever we are talking about soil bacteria, they can be of different habitats, right? Like we can find a soil sample from India also, we can find a soil sample from Netherlands also, we can find a soil sample from say for example Sahara Desert also, right? But each of the soil samples will be having are collected from different habitats which are like poles apart from each other, right? So obviously whatever microbial profiling we will be doing, whatever microbial communities we will be generating from these different widely varying soil samples, they will also be having diverse mechanisms or diverse biochemical pathways which will particularly help them to strive or to survive or to multiply in such diverse habitats. Okay, so on the basis of them we need to differentiate them, right? So let's see what they're saying over here. The first step in examining the potential of microbial profiling for forensic soil analysis is to determine whether profiles generated from separate locations can be differentiated. So that is the first important factor that we need to keep in mind, that since the profiles are generated from separate locations and very widely situated locations, right? So firstly we need to understand that whether it is even possible to differentiate such widely situated locations. Okay, this includes profiles from different habitat types as well as ones that are quite similar. Forensic soil samples may be contested as being from one location or another, and these two locations might be environmentally similar or in close proximity. So either the two locations can be environmentally similar. What do we mean by environmentally similar? Again, I will highlight this part for you. So environmentally similar means the environmental conditions are more or less similar. Okay, means the temperature, the humidity, okay, the ecosystem, the water content, or the the purity of the air. These will be the environmental factors which need to be similar, or which if similar, then we can say that the environmental conditions are somewhat similar. Okay. Or second option is they can be in close proximity, that maybe the environmental situations or scenarios are different, they are not similar, but they are located in nearby geographical locations. Okay. Many soil-based forensic studies have been conducted with the goal of examining if a technique can produce differentiating profiles. TRFLP, I think I have already discussed what is TRFLP in the previous lecture also. So again, if you have forgotten, or again if you have not yet watched the previous lecture, then please go back and watch it. Okay, restriction fragment length polymorphism, that is the full form. Okay. So this is a kind of analysis again which has allowed diverse habitat soil differentiation. However, the quantity and diversity of microorganisms in a soil sample after crowd electropherograms limiting the technique's resolution. So basically it's somehow the resolution of the technique is getting compromised if we are talking about TRFLP. Similarly, DGGE, again DGG is something which I talked about in details in the previous lecture. So this is again another kind of analysis that we can do with the soil microbes, and it can lead to indistinguishable bands on the gel. Basically gel electrophoresis. Okay. Okay, moving on. So the next subheading or subtopic that we need to discuss in this session is temporal changes in soil microbial profile. So let's see what can be the temporal changes. Known forensic soil samples are virtually never collected at the time a crime occurred. Therefore, the change in microbial profiles over time could affect the association of known and evidentiary soil samples. Because see, this is something very obvious, right? Say for example, a crime or a murder happened in a forest area. So obviously it's not expected that then and there, immediately as the forensic team arrives, first and foremost they will collect the soil. No, isn't it? Because before the soil there are some more important evidence materials that needs to be collected. Say for example, all the evidence that is there on the body of the victim, it can include the hair, it can include the nails, it can include the clothes, or any murder weapon if it's found. Then you have to go and search for the criminal. Maybe the criminal is present nearby only, maybe time has not passed that much that the criminal have fled away from the place. Okay. So these are the things that needs to be looked after at first on priority. Then we will go for microbial profile collection and microbial profile testing. Okay. So there's a time that will pass between like till the soil is collected, soil sample is collected, and that time lapse between the collection and the investigation will actually affect the association of the known soil sample, means which is the control soil sample for us, and the evidentiary soil sample, means which is collected from the crime scenario. Okay. Research has shown that microbial profiles do change over time, sometimes enough to affect classification or association. So because these bacteria, these microorganisms, they are very smart. So what they are doing is they are continuously upgrading themselves, means they are continuously mutating on the genetic level, so that they are able to adapt to any kind of situation. Okay. They can adapt to any kind of extreme environmental situation. It can be in terms of temperature, it can be in terms of O2 CO2 content, it can be in terms of humidity. So they have that capability because they can continuously mutate and change their codes and codons and amino acids. Okay. So as a result, what it will do, it will affect the classification or the association, means basically whatever profiling we are doing, it will be affected. Sorry. Seasonal effects and day-to-day weather phenomena may also have an impact on soil profile association. Ideally, say for example, like during rainy season, or maybe during during times like spring and autumn, there is any time there can be thunderstorms, any time there can be heavy rain. Okay, particularly during the seasonal change time. So that can also hamper the soil profile association. Okay. Ideally, a known forensic soil sample will be collected as early as possible. However, if the environment has changed, it may be better to collect them over a number of days or even longer to account for microbial variation occurring temporarily. Okay, moving on to the next subtopic that we need to discuss in this session, that is spatial differences in soil microbial profiles. So let's see what they're saying over here. Just as known soil samples will be collected at a time different from when a crime occurs, they also may not be collected at the exact spot where an evidentiary item was exposed to the soil, potentially affecting the association accuracy of profile analysis. So that is again very important. So just as I discussed in the previous slide, that it's not possible that the soil sample, the evidentiary soil sample particularly, that it will be collected exactly on the time of the death or on the time of the crime or accident, whatever, isn't it? So just as it is not possible to maintain the time, similarly it is also not possible to maintain the accurate spot or the exact spot where the incident happened, isn't it? And even say for example it's just a distance of 10 meters or 20 meters, say for example the spot was 10 meters in front or 10 meters behind, but that 10 meters can actually hamper the profile analysis. That is what they are saying over here. So it's a challenge that it's a challenge to collect it at the exact spot and at the exact time. It is usually not possible. Okay, moving forward. Microbial profiles can be impacted by several microenvironmental factors, including pH, foliage density, nutrient supply, all of which might vary across a single habitat. So again, this is important. So I would like to highlight this part, single habitat. So these are some of the properties, or rather functions, factors that they are talking about. So number one, pH, number two is foliage density, and number three is nutrient supply. So these are three such factors, obviously microenvironmental factors as they are mentioning over here, which can actually vary across a single habitat. So just imagine, even if the habitat is not changing, then also the microbial profile analysis can change or can get affected, right? If we do not consider these few factors. Moving forward, similarly, an evidentiary soil sample stemming from a burial may be a mixture of soils from various depths. Microbial populations are known to differ with depth, likely due to oxygen availability and the chemical composition of deeper soils. So what are the two factors due to which microbial populations can differ? Number one will be oxygen availability, and number two will be chemical composition of deeper soils. Deeper soils as in soils which are situated deep inside the field. Thus, associations between known and evidentiary soils could also be affected if depth is not considered when collecting known samples from a crime. Moving on to the next important subtopic that we need to discuss. So as we can see, it's soil sample collection strategies. So there are some strategies, or rather we can say protocols also, or procedure also, that we need to follow in order to collect the sample for soil sample for microbial analysis. So let's see what they're saying over here. Strategies for collecting known soil samples are exceedingly important to ensure a representative microbial profile can be generated for comparison to evidentiary soil. A single profile may not be an accurate representation of an entire location, and it may not provide enough information to confidently classify an evidentiary soil sample to an origin. Further, both ordination and supervised classification techniques require multiple samples to produce associations between soil profiles. Given this, multiple known samples should be collected from each location in question, as this allows for the formation of representative clusters and training sets during analysis. Further, ad hoc sampling strategies may be needed depending on the specifics of a case. Ad hoc matlab additional, addition basically. Okay, I will just highlight this term. Ad hoc sampling strategies means something which will act as a extra supplementary or accessory factor. For example, the collection of multiple samples branching outward from a main site might be beneficial. However, some locations could have obstructions preventing soil collection in certain directions, for example, a body of water or road. Okay, moving on to the next important subtopic that we need to discuss about in this session: evidence storage and changes in bacterial abundance over time. So let's see what they're saying over here. Unknown forensic soil samples will most often be collected from evidentiary items that have been stored for some period of time prior to discovery. Given this, it is important to understand how soil microbial profiles may change upon removal from a habitat, as well as what affects the evidentiary material itself and environmental factors like storage temperature have on the microbes. Research has shown that bacterial and eukaryotic populations in the soil change when stored. However, soil association is largely unaffected. Predictable profile divergence on stored items suggests that evidentiary soil may have the potential to be used as a biological clock, indicating how long the soil has been removed from a given location. Similar to how insects can be used to estimate a post-mortem interval range for a body, soil could become even more probative if such a biological clock can be developed based on microbial abundance changes. Moving on to the next important subtopic that we will be discussing in this session: costs of next generation sequencing of forensic soil samples. So if we go for this NGS techniques, which is obviously very recommendable nowadays because of high accuracy, then what will be the cost? Let's see. The cost of next generation sequencing has fallen rapidly since 2008, faster than Moore's law would predict. Moore's law was given in the year 1965, so it's a very old law. Again, I will just highlight this: Moore's law. So even surpassing the prediction of Moore's law, the cost actually fell down. The US National Human Genome Research Institute, this is the link that I have given over here. You can just go and have a look at this link also. Reported that the price per megabase has dropped from US dollar 10,000 to under US dollar 0.1 in the last decade. So it's a huge fall, right? I will just highlight this. From 10,000 it has dropped to straight away 0.1, that too in last 10 years only. Overall, producing a microbial profile is extremely inexpensive given the amount of data developed. However, implementation of NGS into crime laboratories could be difficult given the sheer amount of data generated. A single Illumina MiSeq run can produce over 15 gigabytes of sequence data, equaling 25 million sequences or more. Moving on to the next important subtopic, that is legal considerations for the implementation of microbial profiling. Let's see what they're saying over here. The microbial profile generation and analysis methodologies developed by microbiologists for soil samples and tailored to forensic science satisfied Sensabox 3 recommendations for such techniques. What are they? Number one, again I will highlight one by one: reproducible data production. Number two, high discriminatory power. And number three, objective data analysis. Further, adopting already well-established methods should make acceptance in the courts a simpler process, as they meet most or all tenets of Daubert, who did the study in 1993, including prior testing, general acceptance, peer-reviewed publication, and known error rates. Given this, and the broad legal acceptance of DNA-based techniques in general, microbial profiling of soil should encounter minimal resistance from the courts. Moving on to the next important topic, that is consensus on methodologies for soil collection and analysis. So let's see what they're saying over here. Microbial profiling of forensic soil samples must be standardized before these methods can be effectively implemented. Ideally, a consensus will be reached on how soil is collected, what organisms and which regions of their genomes are assayed, the optimal profile generation technique, and the data analysis methods that will be used. This will allow experts to acquire consistent and comparable results when analyzing soil samples. Free open source software packages and databases are currently available on the World Wide Web. Then we have R, then we have Keem, then we have Silva and Green Genes. So I will highlight each of them. You can all have a look at these respective links. I have given the URLs too. Okay. Okay. To analyze and classify large microbial datasets. However, too many options might lead to conflicting results across crime laboratories if different programs or databases are used. Large standardization bodies already exist within the Microbiology Committee. So what we have, the Earth Microbiome Project. Again, I have given the link for you. Again, you can just go and have a look at the website. With the goal of amassing microbial data from around the world for shared use among researchers. So I think that's it in this session. We will soon be moving on to the next important lecture of this course of forensic microbiology. Till then, stay tuned. Please like, share, and subscribe ITLS Academy. Thank you.